So today we will see another topic, plant tissue culture. So what is tissue culture? We all know about tissue culture and how it is being done. നമ്മൾ ഫോട്ടോയിലെങ്കിലും കണ്ടു കാണും how we do plant tissue culture. Plant tissue culture is rather simple and easier compared to animal tissue culture. And definition of plant tissue culture is the in vitro cultivation of a plant cell or tissue under some aseptic and controlled environmental condition, giving the supplements, and they are being cultivated in liquid or in solid or semi-solid media, having the nutrients for the production of primary and secondary metabolites to regenerate into a complete plant. So the tissue culture relies on the three fundamental properties of a plant. One is the totipotency. Totipotency, we know, that is ability to differentiate into any tissues, any type of tissues. That is seen in most of the plants. In some plants, we know that when the leaves are cut and we try to propagate into a plant. When we take a stem and try to plant it, it produces a new plant. So it is the inborn ability of a plant cell or tissue to differentiate into a whole plant. Then dedifferentiation. Dedifferentiation means a reversion of specialized cells to unspecialized forms, so that we can compare it with the stem cells. So stem cells have the same property of dedifferentiation, that is unspecialized tissue to form specialized tissues. Then competency to develop into a total plant, a full-grown plant. Then Haberland is considered to be the father of tissue culture. In early 1900s, he proposed the concept of totipotency, and he tried to culture it in right conditions, under controlled conditions, and the callus cultured from tree cambium. Then the nutrient requirements. These are the inorganic and organic supplements that are needed for tissue culture. And apart from that, we use antibiotics because it must be in an aseptic condition. We have to propagate the whole plant, so we have to mostly add the antibiotics such as streptomycin, kanamycin, activated charcoal in the medium. Then other organic supplements forms protein, coconut milk, yeast, malt extract, orange juice and tomato juice, etc. as the growth supplement. Then growth regulators like plant hormones, auxins, cytokinins. Then demineralized or distilled water is needed. Solidifying agents like agar or gelatin to make a solid media. Then pH adjusters. The medium should be around 5 to 6 pH. So it is considered to be optimum for the growth. And for the preparation of the culture media, this is how we prepare the culture media, like preparing stock solution 1, consisting of these type of magnesium sulfate, potassium dihydrogen orthophosphate, potassium nitrate, etc. Then stock solution 2 include these type of chemicals, and stock solution 3 ferrous sulfate, sodium EDT, etc. So this is just an idea about how we prepare the media. Then coming to the sterilization of the media, all the equipments, including the sterilization media and the utensils, every matter should be sterilized before it is being used for plant tissue culture. So for sterilization of the glassware and metallic equipments, hot air oven is used, and incubator or hot air oven is used for sterilization. and sometimes autoclave is also being used. Then the basic requirements for a tissue culture laboratory are equipments and apparatus needed for tissue culture preparation, washing and storage facilities, media preparation room, sterilization room to ensure complete sterilization of the whole procedure, then aseptic chamber for culture, then culture rooms or incubators fully equipped with temperature, light, humidity control devices. So mostly these culture rooms are equipped with air condition and provided with UV light for disinfection. Then observation or recording area well equipped with computer for data entry. Then this is a common tissue culture laboratory having racks for placing these tissue culture tubes. Then equipment and apparatus include vessels and glasswares. All the glassware should be of Pyrex. Large test tubes, flasks, graduated pipettes, etc. are needed. Equipments include scissors, scalpels, forceps, etc. and spirit lamp. Then hot air oven, a pH meter, a BOD incubator, a laminar airflow chamber, that is for complete sterilized environment, a balance to weigh in the nutrients, data collection and a recording room. So these are the instruments that is used for tissue culture. These are the instruments on the left here. These instruments are for cutting the tissue or the plant part for culture, and this is a sharp knife from which we take a small part of this tissue culture, part of the plant for tissue culture. And these are the pipettes and glasswares needed for nutrient preparation. This is the laminar flow chamber in which we do all the procedures like cutting the plant tissue, implanting it into a culture media, etc. All these being done in this chamber to provide a sterilized condition. Then this is the data entry and recording room. And establishment of plant tissue culture. For the establishment, we need collecting and sterilization of glassware tools and vessels, preparation of the explant, surface sterilization of explant. Surface sterilization of the explant is this explant is the tissue or the part of the plant that is being cut off from the parent plant for tissue culture. So this explant should be sterilized before it is being cut to pieces and implanted into a culture medium. Then production of callus from the explant, proliferation of the culture, subculturing of callus, suspension culture. So these are the steps that involve in plant tissue culture. We will see it in detail. First is the explant preparation. So for the explant preparation, we have already mentioned that explant is a portion of a plant body. It can be anything like stem, root, flower, a leaf, anything, which has been taken from the parent plant to establish a culture. So age of an explant is very crucial because it should not be too old, it should not be too young, and so an appropriate age of plant is necessary. Then homozygous plants are preferred, and these are the explants we usually take, like shoots, roots, flower and leaf are used as explants. So this is a surface sterilization procedure of the explant. We can follow these steps like chromic acid, magnesium chloride, calcium hypochlorite, sodium hypochlorite, alcohol, etc. are used for surface sterilization. And for each of these explants like seed, fruit, stem and leaf, there are different surface sterilizations are needed. Then production of callus from explant. So this is the picture of the callus. This is the callus. It is an undifferentiated mass of cell that is formed after we implant or we introduce these explant onto a culture medium. So this is like a wound formation. When we cut off some plant or from the plant, then that part will develop a tumorous like condition, and that we call the callus. So this sterilized explant is transferred aseptically onto the defined media, transferred to BOD incubator. Temperature 25 plus or minus 2 is followed, maintained, and light is necessary for callus production, and callus production is occur within 3 to 8 days. So this is the proliferation of the culture. The callus is well developed. It should be cut into small pieces. Once the callus is developed, we take out the callus from the plant, from the culture media, and we again do the same procedure. We take a sterilized condition, we cut these callus into 10 or more pieces, and each of these pieces will be again recultured in a new media. So that is and they will proliferate into more callus. That is the proliferation of the media. So this is the stages. That is, in 15 days, here 15 days the callus is developed like spores, and on 30 days we can see a mass of cells are developed forming tissue callus, and 50 days we can see shoot formation, and in 80 days leaves are all complete leaves of leaf formation and shoot formation can be seen. Then this is how these this is how this culture will look like after the whole grown plant can be seen after 80 days or more. So after sufficient growth of callus is developed, it should be periodically transferred to fresh media for maintaining the viability of the cell. So always make sure that the callus must be subcultured. So this subculture will be done at an interval of 4 to 6 weeks, and after the maximum growth, the plant can be transferred to a potling soil under required condition, not in a field, under the roof or in a shaded condition itself, and later it is being transferred to the normal conditions.